So the next step to look at in immunocompetence is the activation sequence leading to the overall immune response. So when we look at how it's going to function, it's all about how the two cells are going to react in terms of the non-recognizable antigen for the B cell or the bindable antigen for the T cell, in which the antigenic proteins are going to be expressed within the MHCs, and these MHCs are then going to go about exhibiting distinct features that will allow for activation. The pathway that gets activated is going to be dependent upon where the antigen is being expressed in terms of inside or outside of cell body, in terms of is it an MHC1 activation, MHC2 activation, MHC3 activation, so on and so forth within the body.
Surface. Thus, the B cell becomes an antigen-presenting cell. If an activated helper T cell has the same specificity as the B cell, it can dock with the B cell and assist it by releasing cytokines. The cytokines trigger the B cell to divide, producing a clone of cells that differentiate into plasma cells. The plasma cells produce identical antibody molecules specific for the antigenic determinant that was displayed by the first B cell, and by the macrophage that activated the first helper T cell. The humoral immune system responds to every antigen in this manner, but different antigenic determinants require different specific helper T cells and B cells.
The bloodstream with antibodies that can bind to the antigen involved in this infection. Antibodies bind to the antigens on the surfaces of the pathogens, marking them for destruction by macrophages. Some of the activation of the immune response typically begins when a pathogen enters the body. Macrophages that encounter the pathogen ingest, process, and display the antigen fragments on their cell surfaces. Macrophages with antigen fragments displayed on their surfaces are called antigen-presenting cells. An antigen-presenting macrophage interacts with a T helper cell that can recognize the same antigen. During the interaction, the macrophage releases a chemical alarm signal called interleukin-1, which stimulates the T helper cell to secrete interleukin-2. Interleukin-2 causes the proliferation of certain cytotoxic T cells and B cells. The immune response from this point follows two paths, one using cytotoxic T cells and one using B cells. Normal cells of the body that become infected can also digest some of the pathogens and display antigen fragments on their cell surfaces. The body makes millions of different types of cytotoxic T cells. Each type is able to recognize a particular antigen. The cytotoxic T cells that are capable of recognizing the antigen displayed on the surfaces of infected cells bind to the infected cells and produce chemicals that kill the infected cell. Death of the infected cells results in destruction of the pathogen.
So now that we've seen what happens, let's take a look at the two ways in which the system is going to go about being activated, in which we have an acute response and then a chronic response. So notice the difference in time between the acute and the chronic is that 72 hours, the 3-day mark, in terms of what's going to trigger a chronic adaptation versus an acute response. And all of this gets linked back to sales gas and the adaptations that take place in terms of the alarm phase versus the resistance phase versus the exhaustion phase, in which the end result of any type of immune response leading to disease or illness is the point where we've reached exhaustion, where we can stipulate that illness and disease is the end result of the neuroendocrine response to stress, inflammation, and pathogenic integration. So let's take a look at the activation sequence. So the activation sequence is going to begin with the pathogen entering into the body. Since it's new and novel, what it's going to do is it's going to excite the NK cells. The NK cells will become cytotoxic and it will induce an endocrine response. The endocrine response will cause B cell activation. At the same time, it's going to cause the alarm phase to take place within the cell's gas response, leading to fluid shifts, which is the fluid shift is going to induce the cardiac shift and cardiac drift episodes within the cardiorespiratory system. It's also going to start to induce a metabolic shift via cortisol response. Within the B cells, what's going to happen is that they're going to become clonal. This clonal B cell activation will release the immunoglobulins. The immunoglobulins, as well as cytokines, will initiate next step within the pathway. The next step within the pathway is going to involve the activation of killer T cells. The killer T cells will then go ahead and take care of the cytotoxic response to the pathogen, as well as lead to activation of macrophages. The activation of macrophages will lead to increased inflammatory response, as well as localized tenderness in areas of active infection. At the same time, anything that's remaining following the active response will become memory. This whole process here is a global response. This global response will become chronic and systematic regardless of the length of actual activation. So even if it's less than 24 hours, if it happens to be a new novel pathogen, we will have this response. It may last less than the 72 hours, but it will still be considered a chronic global response. This is different than when we have a known pathogen coming in. When we have a known pathogen coming in, the memory cells remember the encounter. They recognize the pathogenic antigen. Since they recognize the pathogenic antigen, what happens is it automatically activates the killer, and it signals for the B cells to become active through interleukin 2. The interleukin 2 stimulating the B cells leads to B cell clonal, production of IgGs, cytokine and IgG release into circulation, leading to the activation of further T cells, tagging the pathogenic antigens with IgGs, initiating macrophagic activity, and causing the same type of response that we saw in terms of localized inflammation and tenderness. Notice this is more of an acute and focused response as opposed to the global response. You may still have a sickness or an illness that develops during this phase. However, it's not going to be the same level of response that you see with the chronic response, where you don't have all of the normal flu-ish responses that you have or the feverish responses that you have when you have the new novel interaction. So from these interactions, we're going to be able to develop immunocompetence. That is, we're going to be able to train the B cells and the T cells. There are two means by doing this. One is passive, the other one is active. They're both acquired. Passive acquired activation is what we call vaccination, whereas active acquired is immunization. Immunization is survivorship. So you get exposed to the novel coronavirus, the COVID-19. If you survive that exposure, you're now immunized to that. However, if you go to the pharmacy or to your physician and you get the flu shot, you're being vaccinated for the flu. You're not actually surviving exposure to the flu. So when we think about this, we have to remember is that vaccination is what we get from a vaccine to a pathogen, whereas immunization is what we get from surviving an infection. This survivorship immunization is what gets passed on through epigenetic modifications and genetic modifications, where we have immunizations against distinct pathogens because of survivorship from our ancestors. Which takes us to the idea of what vaccination actually is. So what vaccination is doing is it's training, but goes around that, the acquired immune response, immunocompetency to distinct pathogens. Basically, we're teaching the immune cells how to go about quote unquote attacking. By doing this, we limit the likelihood of having global response, which means that we limit the. So let's take a look at herd immunity. Note the indicated colors here. So we start with a population that has no vaccinations, such as what's going on right now with the novel virus. We introduce people who are infected, and all of a sudden, watch what happens to the population. We start with the same population, but now we have a few people that have been vaccinated. We introduce the same 2 individuals into the population. With some vaccinations, notice what happens to the rate of transmission. Now what happens if we have a high vaccinated population? Notice there is no transmission within the population, but we did not eliminate the pathogen. There's still 2 people who have the pathogen. All we've done is eliminate the means by which that pathogen can be transmitted. So how does herd immunity actually work? Follow the red lines and note the transmissions. And note what happens once we hit that 90 and 95% threshold versus the 0 like we have right now, 50 and 75. Which takes us to a question that we should all ponder as we go into our medical profession. Why would people choose not to vaccinate?